theme of our third and final panel is the new wave of biotechnologies, and will be moderated by Mel Billingsley with Life Sciences Greenhouse of Central Pennsylvania. So Mel, it's all you. Thanks for being our moderator today.
Well, great. Well, thanks a lot and welcome all to this last panel, the new wave of biotechnologies. And I guess in keeping with the theme, the crisis develops accelerated innovation like no others. I'm reminded of the fact that several of the current therapeutic applications being used in the pandemic actually arose out of the last Ebola outbreak in 2014. So remdesivir came out of that, and the monoclonal antibody neutralizing cocktail also came out of that. So necessity being the mother of invention, it's good to hear what the next wave is. So we have an outstanding series of panelists that are going to fill you in on some of their research, all in the general central Pennsylvania region. I'm simply going to moderate and make sure the trains run on time. And we have today Nikolay Dokholyan, a professor at Penn State, whose work is in disordered proteins and using computational methods to understand them as causes of disease and also remedies. We have Dr. Tom Caltagirone of Aptagen, a biotech company outside of York, and he'll explain to us about the use of aptamers, which are a novel nucleic acid synthetic formulation that can be used as both sensors as well as drugs. We have several presentations from our friends at Penn State Material Science and Engineering. We have Jim Adair, who can talk to us about his advances in biomaterials, Jian, who's going to talk about biocompatible materials, and then finally Dan Hayes, who has a series of adventures in biomaterials and will regale us and bring us in. And I would also like to remind everybody that at the end of this at 5 o'clock, there'll be a virtual networking event with virtual drinks. So you're all invited to virtually enjoy. So please check your list for that at 5 o'clock. And so without further ado, I'd like to introduce Dr. Nikolay Dokholyan, and he's going to talk to us about molecular engineering and translational research. Take it away.
Sorry. So thank you very much for inviting me here. It's actually a really fascinating set of discussions. And what I would like to bring to the table is how molecular engineering really can advance translational research. And if you go to the next slide, yeah, so what is translational research and why it's critical to have individual components lined in together. So we have a clinical, translational research in health has particular applications is to human health. It can be in many other directions, but in health, what's important is to connect clinic to science and engineering. So with clinic, we get raw data, we try to understand the symptoms, we try to understand disease outbreaks, and then we want to understand this data and create a theory of what is happening with that. And that requires a lot of experiments and going back and forth to clinic and back to science. And then of course, if we get the theory, then we can actually move forward and actually make some engineering of drugs or diagnostic tools or biomarkers or devices. And all of that will feed into the developing better clinical outcomes, developing better medicine. So that's basically where what is the emerging translational science. It's always been the goal, but it's never been as connected as it is right now. Next slide, please. But the challenge is, frankly, is that you have to have, it's very hard to do everything in one lab. I mean, I don't have an MD degree and I cannot see patients. I don't see patients, but I work with a lot of clinical data. And but in order to understand what is happening with the human at age, let's say 70, I need to dig down back to the beginning of cells, that what happens to cells and what happened to proteins that affect the cells and what happens to interactions within the proteins that actually breaks this protein and affects cell phenotype and then results in organ failure and actual disease. And it actually later manifested the population or at the population level. And so you can see the huge disparity in the length scales. We are talking about between 10 to the minus 10 to actually 100. So it's 12 orders of magnitude if we want to even understand the disease from the basic fundamental principles. And so the translation, that's the major challenge of translational science, is to be able to connect the scales, both in time and length scales. And especially I'm focusing on neurodegeneration, and neurodegeneration starts happening as you are born, but manifests itself at age 60. How do you connect all of this? And how do you identify which person will have likely chances of developing Alzheimer's, and what are the ways to prevent from that? So in my lab, we have been tackling those kind of problems. Next slide. But focusing on molecular engineering. And what does it mean, molecular engineering? And I'll just give one example, and of molecular engineering. There are many different types. So the one which I'm preoccupied the most is the protein engineering. So basically rewiring or redesigning proteins that exist in our body so that they will do something else. What does it mean? So proteins normally, so if you look at what they are they have a linear sequence. That's what's encoded in the gene, and this linear sequence, it's actually becomes a three-dimensional structure, and that's what it's depicted here. So this three-dimensional structure has unique functional properties. So if I change the coloring here, so I take these colors and I change them completely by retaining the same shape but altering their function, I can actually manipulate protein function. It's actually really cool because, please advance. If we re-engineer, for example, here a protein which when we activate, turns out this protein results in a cellular phenotype that is what was very puzzling in cancer biology, because it's actually associated with metastasis. You see, it's called polarized spreading. You can see the cell starts forming a polar structure, and it actually starts to move in one particular direction. So we can actually not only detect, we actually discovered this phenotype using this protein, but we can actually manipulate cell life using these proteins by just re-engineering this sequence. Next slide, please. So, and one of applications that we have had before I came to Penn State, I came here in 2018, was we were working on developing HIV vaccine. And the complexity is so high that it's very hard to find a spot for an antibody to grab, because the protein, the HIV code protein, is covered with sugars. You see these pieces? They are covered with sugars, and because of the sugars, the antibody cannot reach the code of the protein and cannot inactivate the virus. However, it turns out there are certain regions on HIV virus code that's called cone regions, the yellow, orange, and purple, and cyan, that turns out to be actually missing one or two sugars typically in population. So for us, it created an opportunity to actually target those areas using molecular with antibodies. So how can we trigger immune response to orange piece only? That was a big challenge. So what we did, we used molecular scissors where we cut out this piece, here it is, and then we plaster it to a totally different protein, small different protein, which doesn't have sugars or anything. And then we made it very stable. Then we made sure that this protein is expressed as well as monomeric, it's thermally stable, it has nice structure. And then we inject it into rabbits, generate antibodies, and then check whether we can neutralize the live virus with this, with immunizing this epitope, which is not HIV protein, but it has a piece of HIV. And whether the generated antibodies can kill the virus. And turns out they can. And we actually show that, in fact, this is pre-immunization, this is post-immunization. We can actually, and there are lots of controls here, but it shows that we can actually inactivate live virus. Unfortunately, this is not a solved problem yet. We're still working on it. But one of the major, but we deactivate live virus. But the major breakthrough to us was the ability to design proteins so precise when we solve crystal structure and overlay it with what we designed computationally, you cannot see the difference. There are two structures actually overlaid here. There are two structures overlaid here and two structures overlaid here, and you will not see the difference. The difference is so small, it's like, it's less than a meter if you were doing a surgery on a planet 10,000 times distance from Earth to Sun. That's how precise this computation, that prediction was and the design was. And so that ability to design proteins allows us to actually manipulate proteins in real life, in actual settings. If you go to the next slide, please. So when COVID hit us, we decided to apply exactly the same strategy to COVID vaccine. And luckily for us, fortunately, there was less sugars on COVID. And so this is unlike anybody else's strategy for developing COVID vaccine. So everybody else is just randomly targeting various parts of the fragments of the protein. We actually isolate regions that are conserved, which means they are critical. Nature conserved them. It means that they are critical for nature to maintain the structure. And so then, and they are not covered by sugar. So we're actually looking for holes on the surface of this virus that have very important tickle parts that we can attack. So then we transplant, we do the same transplantation to totally, so these are the epitopes that we identified, 1, 2, 3, and we transplanted to totally benign protein. Next slide. And let me just, and then basically, I don't have a slide for that, but we actually have succeeded from preliminary results to trigger immune response in rabbits. And so, and the proteins that we designed are stable. We just learned that the crystal structures diffract really nicely. So if all of this works and we can actually inactivate virus, we're actually talking about vaccine for COVID-19 right here in Hershey. But the kick here is that we can also do diagnostics and prognostics. And this has been a collaboration with several labs in campus and outside of campus, including Erdem Tabdanov at our and pharmacology, Elizabeth Proctor at neurosurgery and pharmacology, and Kirill Afonin at North Carolina, Charlotte. And the idea here is we're basically re-engineering a human ACE2 protein to make it super stable. And then we biotinylate it to conjugate it with double-stranded DNA with quantum dots. The quantum dots is what you see in quantum dot televisions by Samsung that cost thousands of dollars because they're so bright and so cool. The quantum dots are so bright that we can actually use just few of them to connect to our MACE2. Now, this re-engineered MACE2, it will bind to COVID. So then you will, if it binds to COVID, because it's multivalent structure, it has multiple arms, it will basically aggregate. So we called it aggregator, and you will see a signal, a lighting up if the COVID is present in the test tube. So this is what we are doing right now. This protein is almost complete. This quantum dots with this DNA structure is almost done. So we actually will be ready with the test that would be taking 5 to 10 minutes in regular settings. You don't even need, it's like a pregnancy test. So it's gonna be super simple. So next slide, next advance, please. another way to go about is to look for a different perspective, is to actually use our designs, scaffold designs that from the virus to bait for the antibodies. So basically we can actually try to detect whether the person has antibodies to HIV virus by just having this, these epitopes attached exactly the same way as before. So you have the same platform that actually exists and validated to just attach to different engineered proteins. Please advance. What's also cool about all of this is because we can actually detect not only antibodies, we can also detect cytokines. With Elizabeth Proctor, we can profile cytokine response and make early detection available for people who have very small viral load, A, and B, they would have, we can maybe make a prognosis who will get it hit hard and who will have a very mild form of COVID. So this work is in progress, and this also is, I mean, this part is complete. We should just connect the dots here.
1 minute.
So that's what we have not ever stopped working in our lab. We got down to maybe 90%, but we have not stopped because those projects have been going on. And the last slide, please. I just want to mention that the ability to engineer proteins has enormous potential. And one of the biggest potentials I feel like in cancer is CAR T therapy, chimeric antigen receptor therapy. And the idea is actually ridiculously simple. You collect the T cells and, you collect the lymphocytes from the patient. You basically re-engineer T cells so that they have antibodies attached to the T cells so that they recognize cancer cells, an antibody to specific receptors expressed on cancer cells. This way, basically, you then re-inject those cells back into the patient, and then let the nature take its course and destroy the cancer. With CAR T, not only cancer has been treated, but also HIV. There are 2 patients who undergone full recovery from HIV, full recovery from HIV disease. But the problem is that because this reengineering is complex and you can result in destabilizing of the proteins, you can actually trigger side effects and alternative immune response, and it's actually quite a big challenge. And that's what we have been working on with a group at UNC Chapel Hill, is to make next generation CAR T therapy. And I'm gonna stop here, thank you.
Very exciting. So hopefully the participants can see the range of opportunities that are presented by these molecular engineering approaches that are making a huge difference. Up next, we have Tom Caltagirone from Aptagen. Tom, take it away and explain to us the use of aptamers. Tom, are you on? Unmute.
There we go. There we go. Thank you. Thank you, Mel, for hosting this this panel, and thank the rest of you for participating in this event today. I'm the president and CEO of a biotech company, small biotech company in Jacobus, Pennsylvania. I started this company back in 2004, 16 years now. And next slide, please. So actually, this is, I have some, apologize, I have some old photos here. We have to update this. We have an active internship co-op program, but we've advanced to the level that now we have some PhDs on staff, but we still have the program to identify talent, junior scientists that we train into senior staff members. As I said, we're located in Jacobus, been in business for 16 years. We develop aptamers. Next slide, please. So aptamers are sometimes referred to as synthetic antibodies. Here's a white paper that we have published a couple of years ago. on a review of the field of aptamers. They were invented back in the early 90s, so it's been over 25 years now, 30 years now. They're not protein-based like antibodies, they're nucleic acid-based. So they're either single-stranded DNA or single-stranded RNA that bind to targets. Targets can range from small molecules to protein biomarkers to cells, viruses. And like antibodies, they can be used as molecular tools in diagnostics and also tools in drug discovery. Next slide. So here are some common secondary structures or shapes of aptamers that form. There's an aptamer against HIV reverse transcriptase, has a pseudonaut configuration. There's a classic aptamer against thrombin. It takes on the form of a G quadruplex. Another one, a simple hairpin, T4 polymerase, stem loop bulge for an ATP aptamer. These are typical shapes based on single-stranded DNA or RNA. Next slide. So the typical size an aptamer is a fraction of the size of a typical IgG antibody. So they're typically around 30 kilodaltons in size versus 150 kilodaltons of an IgG. And because of its small size, it's a great ligand or a macromolecule for biosensing applications and also to penetrate tissues versus a bulky antibody. Next slide, please. So typical affinities of aptamers are in the picomolar, nanomolar range. The affinities are comparable to that of antibodies, but where aptamers outshine antibodies is in specificity. A classic example that I use is theophylline versus caffeine. Two small molecules, they only differ by a single methyl group, just a few atoms, and yet this aptamer binds to theophylline with 10,000-fold greater specificity than caffeine. And by the way, small molecules are considered difficult targets for any type of ligand or platform, including antibodies. So this is something that many of our customers that come to us have tried the antibody route and have failed for one reason or another, and it's usually because of cross-reactivity or off-target effects. So the next characteristic or advantage of aptamers is that they can develop against unknown biomarkers, and that's the way, because of the way that they're developed, they're enriched from a library or a pool. So you don't have to know the identity of the target in that sample, whether it's a biological fluid from a patient sample or not. And you can use that as a pull down assay to identify the biomarker itself. So it's quite powerful in that respect. Manufacturing, pennies on the dollar, a fraction of the cost of producing antibodies, because these are chemically synthesized using phosphoramidite chemistry, standard technique that's been around for decades. So there's no batch-to-batch variation from one synthesis to the next, unlike antibodies. Stability, that's the other concern of many of our clients that approach us. Because it's DNA or RNA, you can heat denature it and it folds back into shape when you cool it down, unlike a protein-based antibody. So shelf life is important for a lot of these downstream applications using these biorecognition elements. On our website, we have something called a Apta Index. It's a searchable database of aptamers that have been published in the literature, over 500 entries. If you're interested, we provide this as a resource to the scientific community to see if an aptamer exists. It's not an exhaustive list compared to a literature search, but it's something that we live, eat, and breathe aptamers, so we provide that as a resource. But our business model is like a CRO. We develop aptamers, unique aptamers, for our client base, ranging from small biotech companies to academia to big pharma like Pfizer, Merck, Genentech, organizations like the CDC. Next slide. So with any biorecognition element, including aptamers, you can use it in a variety of platforms for detection, lateral flow, like a paper strip test. And we've developed something called an Aptabeacon, where there's no, the aptamer binds to a target and reports a fluorescent or colorimetric output. There's no washing, there's no capturing like in ELISA sandwich-based assays. You just read. Next slide. You can also use these aptamers for targeting tissues for imaging potentially. Here I have some internal data that we've generated where we've enriched an aptamer or an aptamer library against lung tissue. This is data from real-time PCR or quantitative PCR. After 9 rounds of enrichment of a library targeting lung tissue, the polyclonal pool binds to lung with a 10-fold signal to noise ratio compared to the background tissues that we tested. From that pool, next slide, we've identified a monoclonal candidate, and this is fluorescent microscopy imaging of a cross-section of tissue, testing the panel of tissues were negative under the scope. This aptamer was labeled, by the way, with a fluorescent tag, and the lung lit up compared to the panel of negative tissues. We saw some background fluorescence in liver, but we had to crank up the exposure. Well, actually, yeah, I apologize. That the background fluorescence could be due to one of two reasons. One, there's a common epitope expressed in liver versus lung, or the more likely scenario is that in liver, these are nucleic acids that end up eventually being cleared from the kidneys or liver, and these are the byproducts from that clearance mechanism. But we had to, as I was going to jump to, we had to crank. The lung is twice as bright as the liver, so there's specificity in the lung. And this is just I'm just giving a brief snapshot of how we use these aptamers for our clients. We target cells. We target many times we get contracted for small molecules, which are considered difficult targets. So with that, I'll stop here. I guess we'll take questions at the end of this panel discussion. Thank you, Mel.
Hey, thanks a lot, Tom. So now you've heard about proteins and nucleic acids as probes. Now we're going to go into the different realm of biomaterials, and with that, Jim Adair from Material Science and Biomedical Engineering. Jim, take it away.
Thank you. I'm going to talk a little bit about the kind of collaboration that we do here at Penn State. It's rather unique in my experience, and I've been at 3 different universities. Penn State has all of the tools built in to foster collaboration that most academic institutions do not have. Could I, the next slide, please? So in 2003, I began working on the notion that we could create an inorganic, biocompatible, bioresorbable drug delivery system. And it turned out that during the development, that if we add a small amount of silicate to calcium phosphate, we can stabilize the amorphous phase. which gives us the advantage of being able to put a bunch of small molecules on the inside of the particles. Can you see any pointer or anything? No?
The arrows, yes.
Okay, good. Thank you. So in this case, we can put both imaging agents and therapeutics. And along the way, we discovered with arguably the oldest near-infrared imaging agent, indocyanin green, that we could also elicit an immune effect, heretofore completely unrecognized, because in the free state, it doesn't generate an immune effect. Inside our particles, it does. So these particles are nominally about 10 to 20 nanometer. We're getting pretty good at pretty much sticking around 10 nanometers for our smallest formulation, but the particle size is tunable because we make these in reverse micelles. And most recently, the National Cancer Institute at the NIH Nanotechnology Characterization Lab has accepted the, next slide. So we have lots of collaborative teams that we've worked with over the years. Most of these are Penn Staters. Several hundred, in fact, depending on the nature of the project that we've been working on. Next slide, please. So this is a group that was really headed by Gary Clausen, and Tom's talk was very timely, so I don't have to go into detail about aptamer targeting. He already kind of set things up very nicely for me. Thank you, Tom. So we compared the co-localization of our nanoparticles that contained indocyanin green with an aptamer target very specific to the CCK beta receptors that are overexpressed in all human pancreatic cancer. And what you're seeing here is the aptamer. It's very bright, much brighter than the exogenous agent. We only used a relatively small polypeptide. The gastrin that's got receptors on the human pancreatic cancer, the CCK beta. As Tom indicated, we found that the association constant was on the order of nanomolar, whereas this aptamer was picomolar, about 1,000-fold stronger interaction than the exogenous agent that's a natural part of the progression of pancreatic cancer. We're also able to pick up some of the metastases in these orthotopic cancer models. We can also follow the clearance of the particles. The calcium phosphosilicate, regardless of bioconjugation, and we put half a dozen or so different, well, probably more than that over the years, but a number of different target molecules onto the particle surface to light up various types of cancer. Breast cancer is one we're currently working on, particularly metastatic breast cancer. So this particle is bioresorbable, biocompatible. It's got a built-in trigger for its release of the encapsulated agent when the particle undergoes endocytosis, particularly the targeted endocytosis, and gets incorporated in this vacuole within the mammalian cell. The endosome is the recycling center for the cell, and enzymes whose maximum activity is 4 to 5 get associated into the endosome, and at pH 4 to 5, calcium phosphate dissolves. Yet some of us inflicted this notion on our children. You take the baby teeth and you put it in Coca-Cola overnight, it literally does dissolve at the pH 5 of your typical Coca-Cola. So this has most of the attributes that you want, except for one until fairly recently, and that's scalability. We've developed a scaled up process now where we can make basically silo levels of material. Next slide, please. Next slide. Thank you. So let's look at the situation with the current pandemic. And it's a little cynical question. Why was the world unprepared for SARS-CoV-2 pandemic, as it was unprepared for the Ebola epidemic back in around 2013, 2014? Well, we've depended on at least respiratory virus in particular. We've depended on annual vaccines. And it's kind of like when I was a kid, got my first car. It was the first, around 1968, they started putting seatbelts in cars. And of course, at that time, we had something like 50,000 deaths on the roads in America every year. And that, of course, has dropped down tremendously over the past 50 or 60 years. Well, this is data from the CDC. Over the last 10 years, the last 10 years of flu season, not counting the 2019-2020 flu season, every year, the minimum number of illnesses was 9,000, up to 45,000. That was 2017-2018 flu season, just about 2 years ago. The hospitalizations ran between 140 and 810. This is all US data because it's from the Center for Disease Control. And deaths were 12,000 to 61,000, with the 61,000 being for that very bad flu season, which, by the way, us old people escaped it, and it actually hit the younger patients between 18 and 36 in terms of morbidity. Now, since the 70s, the state of care has been vaccines. Some people take them, some people don't. I'm absolutely religious at taking my vaccines because I'm getting old enough to realize I am definitely not immortal. Vaccines usually require 18 to 24 months to develop. And this is a bit of a problem because the virologists and the healthcare experts have to try to project out what particular protein are they going to develop the vaccine for 18 to 24 months away. And they often fall a little short, understandably. Now, viruses have built-in ways to avoid the immune system. They're a foreign particle, but they can avoid the immune system. One way, the SARS-CoV-2 can avoid the immune system because it's a little bit on the small side. Phagocytes tend to recognize particulate matter that's 300 nanometers. And of course, the single SARS-CoV-2 is around 120 nanometers. The influenza viruses have glycoprotein surface molecules that promote phagocyte recognition avoidance. For example, hemagglutinin not only promotes the avoidance of the immune system, but it also promotes the endocytosis for the cell through the membrane pits in the cell membrane. And also, viruses generally don't respond to the typical bacterial antibiotics. Possible exception, coronavirus, but right now the waters are so muddied, it's hard to say what will work and what won't. But we'll slowly figure that out as we move away. So, oh, next slide, sorry. So we put together a team in mid-March. It was in response to a DoD call for therapeutics, among other things, in the wake of the crisis that we were facing. Joyce Jose is the main person who did this work. My group, of course, made the nanoparticles. Susan Haffenstein is also a virologist along with Joyce, a structural biologist who can model and render changing and very specific protein modeling on the surfaces of various virus. Gail Matters is a incredibly good biochemist at Hershey Med in pharmacology, even though, well, she's in biochemistry and molecular biology. Scott Halstead is a pediatric pulmonologist and a PhD MD with the particular expertise in immunology. Deb Kelly is a protein structure person, and then there's me, and God only knows what I do, except make nanoparticles. So we developed this treatment, the provisional patent still pending, that I'm going to call targeted therapeutic nanoparticles. And Joyce added, we didn't know how many particles, we just freshly made this new material, or even how many particles were in the untargeted, that would be here. But this was the result of this plaque reduction assay that a lot of people use to identify the SARS-CoV-2 number concentration, as well as for other viruses. And after 60 minutes, after we injected the tRNPs into the SARS cell culture, we got 90% knockdown in the population of the number of virus. Started at 100,000 per ml. By the way, that's a rather modest concentration. At the height of infection, you can have as many as 10 to the 8 particles. Now, with our particles, we're typically at 10 to the 11th to 10 to the 13, more than ample particulate. And these are all 10 nanometer basic sizes. The percent viability of the SARS was knocked down to 25%. This is one data point. We have a lot of work to do. We have 2 other, this would be strike 1 in our formulation. Strike 2 and 3 are on the way. In the meantime, we're working on evaluating the effect of these particles on Zika virus, influenza, because these are quite general. And I fully endorse what Tom said about the certain proteins are conserved from one generation to the next in viruses, and those are the ones that you really want to target. So our next step after this proof of concept.
Can we about a minute to go, we want to get the other couple.
I'm wrapping up. There's the last 2 sentences. So strike 1 is the proof of concept demonstrated. We're going to broaden this out for this particular type of neutralization to other viruses. The next steps are proof of concept for strike 2 and strike 3, along with the validation, which includes the dose response. And I will stop here.
Excellent. Thank you very much. So you can see the power of nanoparticles. Small, big things out of small particles. Next, Jian is going to tell us about his work in biomaterials to look at functional biomaterials.
Thank you, Mel, for organizing the panel. And also, thanks the M and other people organizing the great event. So, so I want to just spend the next few minutes to share with you what we do here at Penn State. So a little bit about myself, and I'm a professor in biomedical engineering, work on biomaterials. I also have a co-founded company in the State College called Adair BME, working on a few biomaterial related technology. So some of the technology I'm going to talk about here also licensed to the companies, in New Jersey, so working on medical device. So I want to share with you how we develop biomaterials and what we see, what would be next generation. So next slide, please. Yeah, so most of our work, we focus on a type of materials called citrate-based biomaterials. Citrate, this is like lemon molecules, right? So it's, everybody knows the citric acid. It's very common molecule, it's very safe, very biocompatible. And chemistry-wise, and it's very powerful, starting chemical that you can build a lot of different materials around citric acid. So over the years, we have built a methodology that, starting with citric acid, carefully select different compounds we can to react with citric acid. We can build a platform biomaterials. So the materials can be soft, can be hard, can be elastic, can be injectable, can be antimicrobial, can be imageable by MRI, by CT, by fluorescence, photoluminescence, things like that. So we have a platform biomaterials, and as you can see that on these slides, we have worked on many different medical applications. Fortunately, I have a lot of connections or collaboration with with many physicians, surgeons in Hershey Medical Centers, working on a number of things, including wound healing, bone regeneration, and like nerve regeneration, and a sensing like sweat test for cystic fibrosis diagnosis, like that. So as you can see, it's a platform can be used for many different applications. But as you know, for biomaterial scientists, people, you can see there's so many literature people working on a lot of different new biomaterials. But a lot of things probably remain as an academic practice, right? So, and the sense that when we do biomaterials, I always have something in my mind that how can we translate this technology, right? So that means if you want to build a biomaterial, you do not want your material to be very sophisticated, and also the compound, the monomers that you use to make the materials has to be safe. So this is why. So we're fortunate that we have built that methodology that we can use the power of the citric acid that can easily react with many different compounds. We select the very a lot of different compounds, has very good safety record to react with citric acid to build some materials. This is why some of our materials has been licensed to some other company for application for commercialization right now. So so next slide, please. So I want to share with you a few different applications. For example, we can make some very soft and elastic materials to make like blood vessel scaffold or nerve conduit for blood vessel regeneration or for nerve regeneration. We can also, based on our citrate-based materials, to make some antimicrobial polymer that can be used for 3D printing. We can print this kind of wound adjacent materials, and we can also make very hard, very strong materials that can be used for bone regeneration. For example, on the bottom left, we're making a long bone regeneration, making a scaffold to help the long bone regeneration. So, and also on the bottom right is something that we work on, the spinal fusion. So basically, this is showing that our materials can really instruct the bone regeneration. So when I say instruct the bone regeneration, we are actually making instructable biomaterials. Basically, because when we work on biomaterials, we do not just simply say the material just a materials for device fabrication. We want the materials can instruct how tissue, instruct tissue healing or cell response, things like that. So this is why. So there are a lot of things with fundamental research we also do around our citrate materials. Next slide, please. So for example, I just want to give you one example. Here we work on bone regeneration. We can make our materials into different orthopedic fixation device like bone screw, suture anchor, spinal fusion disc, or this kind of bone plate. But this is just not a biodegradable materials. This also has a biological implication. For example, we know citrate is a metabolite, right? So it's an intermediate in the Krebs cycle for energy production for cells. And actually, when the stem cells trying to make new tissues, they need energy. So we can use our citrate biomaterials, which is degradable. When they degrade, release citrate that can be uptaken by the stem cells to modulate the cellular energy level. The cellular energy level change will also regulate the stem cell differentiation into bone cells to make bone tissues. This is one example that we demonstrate we can use citrate to mediate the cellular metabolism, which in turn mediate the
10 minutes.
Tissue regeneration. So citrate is not just only for regulating metabolism, it also actually can regulate the local pH to influence or to activate some tissue growth factors to help wound healing. There's so much to do behind the chemistry and behind the materials. There are a lot of biology behind that. So the next slide, one more slide. So here is another example that we are working on the instructable materials. We make very soft and elastic materials, and that we can make this nerve conduit for peripheral nerve regenerations. And but we can make multi-channel this kind of conduit to physically guide nerve regeneration. But what we're doing right now is that we also incorporate folic acid. This is a vitamin B9, and this can be a great source of DNA methylation. Because we are using folic acid. This is a very well-known food supplement. But there's a new mechanism that we can utilize folic acid to regulate the DNA methylation, which also can help to improve the nerve regeneration. So we are doing this kind of new biomaterials, incorporating folic acid into the polymer to slow release or lifetime release folate to stimulate the nerve regeneration. So this is the collaboration with Elias in neurosurgery. So just one example. So this is what we do. So like I said, when we're trying to develop biomaterials, we want to make sure that everything going into the polymer should be safe. And also because I have been working with the companies, going through some of the regulation process, understand how difficult to have a new biomaterial, especially biodegradable polymer, to be used in FDA-approved device, and that we will have some very good news to announce very soon. But I don't want to say it here now. So, but what I'm saying is that it's very important to develop biomaterials, not just only working as a substrate. It also has biological implication, but it has to be simple, has to be low FDA barrier. So that is my personal experience, my 2 cents. So I'm gonna stop here today.
Thanks, Dr. Jian. And last but not least by any standard, Dan Hayes. Dan has quite a background in nanotechnology and materials. Dan, take it away.
Thanks, Mel. So I'm going to talk a little bit about spatial and temporal control of gene expression in vivo using a nanoparticle-based platform. And this is similar to the kind of work that Jim was talking about, but I'm going to talk about instead of targeting particles to a diseased tissue, using an external stimuli to control the activity of those particles in the tissue. Next slide, please. So the system that we've developed is a light-activated system for the delivery of siRNA, or small interfering RNA. Small interfering RNA are RNA that will interact with the messenger RNA in the cytosol to modulate post-transcriptionally the expression of proteins. They're very potentially powerful therapeutic. There's a subclass of siRNA called microRNA mimics, which are mimics of the naturally occurring microRNA that are conserved, evolutionarily conserved, to have distinct biological functions. The one that we're going to talk about today is actually microRNA 148B, which targets over 300 genes in the cancer pathways. And so it's a very, very powerful tool for modulating cancer cell behavior. The downside is that these microRNA also have a lot of effects in other tissues. It targets 300 genes in cancer pathways, but it also targets hundreds of genes in other pathways, meaning that we have to get really fine spatial and temporal control over it, or they're going to have really dramatic side effects. And this is, to date, has really limited the ability of these microRNA mimics to be used as therapeutics. So the system we've developed is this nanoparticle delivery system that is activated by light. We deliver it through IV, it's taken up by the tumor, and it's only activated where we shine a light on the system. So next slide, please. You can see in the upper left-hand corner the schematic of the nanoparticle system. So we use a Diels-Alder cleavage chemistry to link it to a metal nanoparticle that is a plasmonic particle, so it resonates at a specific wavelength of light. In this system, we used a silver nanoparticle that resonates very specifically at a 415 nanometer wavelength, which is a blue light. When the particle is excited using that 415 nanometer light, it generates heat, it generates plasmons and hot electrons, which drives the cleavage of that Diels-Alder structure, releasing our compound. What's interesting about Diels-Alder structures is they can cleave very predictably. So they're stable at under physiological conditions, but when you give them enough energy, they'll cleave and revert to their original diene and dienophile. Makes it very easy to predict what the biocompatibility of the resultant materials are going to be, because they're the same materials that we started with. We make the diene and dienophile into a Diels-Alder, we cleave it back and get the same starting materials. You can see down in the bottom left-hand corner are the actual particles themselves. And if we go all the way to the right-hand corner, bottom right-hand corner, you can see the release profile of these particles. As I said, it's a true covalent bond, so it's very, very stable when we put it into physiological systems. So while it's circulating, we don't get any spurious release. But when we give it enough energy, you get this step-like function of release. So only where we apply the light and apply enough light are we going to get the release of our therapeutic, in this case, our microRNA mimic. Next slide, please. So we demonstrated that we were able to control these intracellularly. So on the left, you can see the nanoparticles themselves, and that's just the plasmonic resonance, the backscatter from it. There's a fluorophore on the microRNA mimic that's attached to the particle, but you can't see it because it's quenched via a fret-like mechanism. It's not quite fret, it's very similar. When we shine a light on it, which you can see there in the middle panel, it's released, diffuses into the cytosol, and you see the distinct fluorescence that comes from our fluorophore on the microRNA mimic. We also demonstrated that we were able to knock down a number of the genes that are direct targets of microRNA 148b, the naturally occurring microRNA 148b, using our mimic. So as I said, there are over 300 targets in the cancer pathway. We picked 5 common ones that we understood their roles well and knew that they were confirmed targets. And you can see that we knocked them all down intracellularly in a cancer cell. So next slide, please. We then wanted to see if it had the discrete biological activity that we had hoped, which was driving apoptosis in cancer cells. So in the top row, you can see normal keratinocytes. In the middle row, you can see human RAS-expressing keratinocytes. This was done in conjunction with Adam Glick at Penn State University, who provided both the animal model and these cells. And then a PAM212 cancer line. You can see they have no effect on the light-activated nanoparticles have no effect on primary keratinocytes, but in both of our cancer-type cells, they drive apoptosis, which is what we were hoping. This is done through an apoptotic mechanism. If you look down at the bottom, you can see the protein expression for cleaved caspase. And in the case of our control or our PBS control, we don't see any activity. In our non-coding control, I should say I didn't mention this, but we're using a non-coding RNA control that is a scrambled version of our microRNA 148b. And then in our actual 148b microRNA 148b mimic, we do see that apoptotic reaction. Next slide, please. So most importantly, what happens when we put it into a cancer mouse model? As I said, these are Adam Glick's squamous cell carcinoma model. It's a human RAS expressing genetic model. In the top left-hand corner, you can see the distribution of the particles when we injected them into the mouse. And in contrast to the work that Jim was talking about, these particles go everywhere. They do not concentrate in the tumor. In fact, a relatively small fraction of them ends up in the tumor compared to liver, spleen, heart, and other tissues. But the key here is that we're using an external stimuli to control the activation. The cells that go into the liver, spleen, and other tissues are not activated. They degrade or are excreted from the system. You can see in B that that's the tumor volume of the tumors in the mice. And we made our injections at day 1. By day 2, the tumors had in our light activation on day 1. By day 2, they'd lost approximately 90% of the tumor volume. By day 3, it was about 99% of the tumor volume, compared to the either non-coding control or PBS control mice where the tumor volume kept expanding. So this was a really dramatic reduction in the volume of the tumor in the areas that we had light activated. So if you look at the mice, you can actually see one of the mice, and that's a 24 hours after the mouse has been injected. So the top mouse is its initial state, and then 24 hours later, the tumor is largely gone. And you can see the histology of the different samples just to the right of that. So the PBS control, then our non-coding control, and then the tissue after it's been light activated with the nanoparticles with microRNA 148b mimic. There's a major degradation of the tissue. The tissue is being rapidly digested. And moving on to the next slide, please. So obviously, given this rapid collapse of the tumor volume, we expected there to be some sort of immune function related to that. And so we looked at a number of immune cell surface markers to determine if immune cells were infiltrating into the tumor volume, and not surprisingly, they were. In the top row, you see our PBS control. The middle row is our light-activated non-coding, and then in the coding, microRNA 148b, you see this massive immune cell infiltration into the tumor volume. Next slide, please. So we wanted to see if there was any sort of polarization specific to the nanoparticle activation themselves. one thing we find is that we do not see this response in the normal skin that surrounds the tumor. So the concentration of immune cells in the tumor is much higher than in the normal skin surrounding it. And that dovetails with what we saw when we did our control experiments with our cells, that our normal cells do not respond to the microRNA 148B mimic, at least do not respond in the same way to drive apoptosis. When we looked at the immune cell function, we found that in the spleen and in the tumor, we had an activation of Th1, or polarization for Th1, and the upregulation of those markers on the immune cells that we were able to isolate. This was easily seen in the spleen. It was a little bit more difficult to do in the tumors because of the rapid reduction in volume and the rapid reduction of tissue and cellular material. But we did see an upregulation in both. Interestingly, we did see some upregulation with just the nanoparticles themselves, regardless of whether or not it had coded for the microRNA 148B. So the nanoparticles themselves have some effect on the immune system, and we're still trying to figure out exactly what that is. We also saw activation of Th17, upregulation of Th17 markers in the spleen in the same pattern that we saw upregulation with just the non-coding microRNA on the non-coding siRNA on the nanoparticle, but then a slight upregulation with the microRNA coding. Next slide, please. With that, I'd like to thank our collaborators and our funding sources. This was funded by NIH and the Department of Defense primarily. Thank you.
Excellent, thank you. Well, hopefully the audience has gotten to see a wide range of emerging technologies. I think that's very reassuring in a time of need. Any questions? I didn't see any coming through the chat because I think we're between the virtual happy hour, virtual interlink. But I'd really like to thank all of the participants for their excellent presentation in the virtual seminar. And it's always encouraging to see new and exciting approaches. Who knows when we'll need them? The next pandemic is always lurking around the bend. With that, I'd like to thank everybody and turn it back over to Anne and to get ready for the happy hour.